Good morning. Welcome to the Facioscapulohumeral Muscular Dystrophy Adjunct Scientific Workshop. My name is Dr. Michelle Mellion, and I am the medical consultant for the FSHD Society. Today's meeting is an opportunity for the FSHD community to share our progress and collaborate with the FDA on clinical trial endpoints. This meeting follows productive interactions with the agency, including a patient-focused drug development meeting held on November 5th, 2020, and a patient-led listening session on upper body mobility on August 23rd, 2024. James Valentine will review the themes and outcomes from the PFDD and PLS. The goals of today's meeting are to discuss FSHD clinical trial endpoints from the patient perspective and for leading experts to provide an overview as well as report on the progress, including clinical outcome assessments, musculoskeletal MRI, and biomarkers. Experts will provide a presentation on the background and advancements of these assessments and potential strategies to address challenges. Our presenters today include Grace Fisher, providing the patient perspective, followed by Dr. Jeff Statland, Professor of Neurology at the University of Kansas Medical Center, Seth Friedman, Manager of Innovation and Simulation Modeling at Seattle Children's, Sylvia Blemker, CSO and co-founder of Springbok Analytics, Per Widholm, Lead Scientist Musculoskeletal from AMRA, and Suja Jagannathan, Assistant Professor at the University of Colorado. The presenters will then be joined by their colleagues for an open roundtable discussion to be moderated by Lucienne Ronco, the Chief Scientific Officer of the FSHD Society. The number of clinical trials in FSHD is increasing, with several ongoing and completed trials focused on different therapeutic approaches, with most focused on the root cause of disease, expression of DUX4, are processes that are closely associated with DUX4 expression. The toxic gain of function due to de-repression of DUX4 causes relentless progression of cranial to caudal muscle weakness, significant morbidity, and early mortality. FSHD has been clinically diagnosed in an estimated 8,000 to 10,000 US citizens. It is a genetic condition which can be either inherited or occur spontaneously. The onset of symptoms can begin at any age, and anyone can be affected. Given the relentless nature of the disease and that the cause of the disease is the same regardless of age of onset or clinical phenotype, treatments for FSHD should not be restricted based on arbitrary inclusion criteria, but rather should be appropriately extrapolated so that the entire community has access to treatment. Several clinical outcome assessments, as well as molecular biomarkers and MRI protocols, have been studied and included in ongoing clinical trials. The progress we have made with these clinical outcome assessments, such as walking tests, timed up and go, and reachable workspace, as well as molecular biomarkers, will be discussed today. Additionally, different MRI protocols have been assessed in this population. This figure shows the baseline assessments and severity of disease shown in different colors of individual muscles. All of these assessments, clinical, biomarker, and MRI, have demonstrated capabilities to capture disease heterogeneity and progression. Now is a critical time for our community to work collaboratively with our regulatory partners on understanding these important clinical trial endpoints supporting the development of disease-modifying or potentially curative therapies. The FSHD Society is a partner working to bring together all stakeholders. The Society is the world's largest research-focused patient advocacy organization for FSHD. For over 30 years, the Society has driven major advancements in research and treatment development for the mission to end the pain, disability, and suffering endured by 1 million people living worldwide with FSHD. The Society supports families affected by this disease through programs like Better Life, which empowers patients with tools to track symptoms and connect with researchers, the FSHD Navigator, a service providing personalized assistance to help handle the challenges of living with FSHD, as well as supports advancements of therapies through the Global FSHD Innovation Hub. Our promise to patients is that as long as we are here, no family need ever face this disease alone. I will now hand it over to James Valentine to provide a summary of the PFDD and the patient-led listening session. James?
Thank you so much, Michelle, and it's a pleasure to be here today. My name is James Valentine. I'm a director at Hyman, Phelps & McNamara, which is a boutique FDA regulatory firm in Washington, D.C., and it's been a great pleasure of mine to be a regulatory advisor to the FSHD Society since 2019. If we can go to the next slide. So we just heard from Michelle a great agenda stacked with leading experts, clinician and research experts in the FSHD field, who'll be talking about important tools for drug development in FSHD. But it's important to the society and to all stakeholders that we ensure that that discussion is patient-focused and builds on the learnings from the FSHD Society and their partners' previous activities. This entire patient-focused drug development, really culture shift within the drug development arena, dates back, I can't believe, already 12 years to 2012, when FDA launched its patient-focused drug development initiative and put on the record that patients who live with the disease have a direct stake in the outcome of FDA's decisions, and importantly, are in a unique position to contribute to the understanding of their disease. I'll spend just a few minutes talking about where the patient voice can fit in to drug development and review, and then share some of the learnings from the FSHD activities. So when we think about drug development, we often are thinking about, well, what is that decision that ultimately allows a product to get to patients on the market? And that's the FDA approval decision. And so as we think about that and approving a new drug, FDA absolutely is making a scientific determination. They're weighing the benefits, assessing the benefits of the drug and seeing if they outweigh their risks. And they need to use their scientific expertise to know what are the benefits, what are the risks, and how much certainty do we have about what those benefits and risks are. However, if you go to the next slide, there is a subjective element to this. When deciding whether the benefits outweigh the risks in that trade-off, it's a value judgment. And so it's important to know that FDA is calibrating the values on how much risk a patient population should tolerate against any given set of benefits by learning and listening to patients, just as they've done with the FSHD Society events. Next, and I just want to make a point here that when we talk about risks, we may immediately think about safety risks, and that is incredibly important to understand risk tolerance when it comes to side effects and other adverse events. But it also relates to risks in terms of uncertainty of treatment benefit. And that's very critical to the discussion today, where we're talking about biomarker approaches, which are inherently not the measure of how a patient feels, functions, or survives. So it's not the inherently meaningful outcome, but it may allow access to products earlier in development to patients. And so uncertainty of whether, or that is an inherent uncertainty, and patients' preferences about accepting that uncertainty is very important to take into consideration. If you move to the next slide, so I wanted to not just talk about that subjective kind of terms, but really share with you where FDA fits this patient input into its own regulatory framework. And so when FDA is assessing the benefits and risks of the product, it considers two categories of the patient experience, which you would have heard about at the patient-focused drug development meeting, and I'll share a little bit about today. The first are those direct experiences with burdens in patients' daily lives. But the second also builds on that and assesses patients' perspectives on the adequacy of available therapies. And this helps FDA understand what types of benefit matter most to patients. And if you go to the next slide, you'll see it very clearly that in FDA's own structured benefit risk decision framework, the first two rows of this framework that they fill out for any particular product that they're reviewing have nothing to do with the product itself, but really relate to those two areas of understanding what is most troublesome to patients as a result of their condition and how it impacts their daily life. And in the second row, what their unmet medical needs are and their treatment goals. If you go to the next slide, what I talked about so far is really what happens very late in development, when FDA actually has all the data from a development program and is balancing benefits and risks. But important to today's discussion, the patient voice is also very important in thinking through the development of products and how trials are designed. And specific to today's topics, that's going to focus on the selection and development of clinical outcomes assessments, which are assessments of how patients feel or function, and they can be either measures of performance, they could be ratings done by clinicians, or even assessments done by observers like caregivers or patients themselves. And so if you move to the next slide, some of the key questions that patient voice, patient input can help inform are what are the right things we should be measuring in clinical trials? Which symptoms and health effects or functions are important to patients or represent greatest unmet needs? How's the best way to go about measuring them? Is there do patients describe a lot of variability day to day where we might want to use a decentralized digital health technology or a patient diary on a daily basis to understand that? Or can we reliably assess it every 6 months at a clinic visit? And then ultimately, an important question is what amount of either improvement or between group difference compared to a control and maybe progression in a control group would be considered clinically meaningful. So if you go to the next slide, so now we want to share with you at a high level, this does not do justice to these events that occurred in 2020 and 2024, but some highlights from these two interactions between the FSHD patient community and FDA. You go to the next slide. So the externally led patient-focused drug development meeting was a wonderful event. There were, I think, over 600 patients and caregivers that contributed in the written survey, and over 300 participants from the patient community who participated in live polling and other live meeting activities. And so on this slide, what you're seeing is a summary of some of the key takeaways from that first discussion about what is most impactful to patients in their daily life. You can see on the left figure on the slide some of the symptoms that are most significant in impacting daily life. We see impaired mobility, fatigue, difficulty using hands or arms as some of those top most burdensome symptoms. And then on the right hand, you can see what that means in terms of how that impacts daily life and some of the activities that are most impacted. You see walking, participating in sports and hobbies, going out, socializing and traveling as some of the top reported there. But more than what these figures can share, you can see in the words of some patients what that impact means. One man said, I wasn't able to pick up my kids out of their crib or throw them in the air like many of my friends did with their kids. I had to sit down as my wife handed them to me, just like she did with their great-grandmother who was fragile and weak. And you can see in the other quote, the 27-year-old woman said, I've fallen by myself many times. And I think one of the scariest things about falling, especially by yourself, is I'm scared that I'm going to hit my head. I'm scared that someone's going to find me unconscious. I'm scared that one day I fall and I'll break something and lose even more mobility. So you hear not only about the impacts, but the worries that patients have to bear day in and day out. If you go to the next slide, you'll now see some of the patient perspectives that were shared around treatment approaches and what their unmet needs are. On the left-hand figure, you'll see that patients rate the effectiveness of current FSHD management approaches, things that are fairly limited, over-the-counter pain medications, dietary and herbal supplements, exercise and mobility devices, being some of the most common management approaches that they have. And you can see that about 42% rated this as helping somewhat, and 41% very little. So a great unmet medical need that exists. And so when asked about what their patients are looking for from future treatments, short of a complete cure for FSHD, 62% rated slowing or stopping the loss of muscle function as the #1 treatment effect. And that's very important. That means even if a treatment were to slow the progression, so they still worsen over time, but at a slower rate, that would still be a meaningful treatment outcome for these patients. And again, you see on the bottom some of these direct quotes talking about the importance of being able to live independently or at least stop progression. And a mother of a young daughter who's 11 talking about the unpredictable and variable nature of FSHD. And so anything that can help make this more predictable and slow those losses being important. If you go to the next slide. Building on this very important patient-focused drug development meeting, there was a recognition that it's much less understood the impacts of upper body mobility. Often when we talk about mobility, we think of ambulation or the ability to walk, but in FSHD, that's not the full picture. And so 96% of people with FSHD suffer from loss of upper body mobility. And this has a huge impact on independence and often can be greater than the loss of ambulation in this patient population. And so in this meeting where a handful of patients and caregivers met directly with FDA, you can see that they shared some of the impacts of this upper body mobility limitation. One patient describing it as, imagine it having rope tied around your arms, tying it to your body. Another talking about, imagine your favorite thing to do in the entire world, whether that's sipping tea, playing a sport, holding your child, and that being stripped away from you. That's what it's like to live with this upper body disability. And so then when describing treatment goals and consistent with what we heard at the PFDD meeting, there was an eagerness for a slowing of progression, knowing that even maybe then existing compensations would last longer. And some did describe how functional improvements could be valuable, but those could be very small, like being able to write or type for 5 more minutes to get homework done, or being able to move their shoulder an extra 10 degrees so they can reach a cup to their mouth. So we ask that you keep this patient voice in mind as you think about the topics and discussion today. And at this point, it's my pleasure to turn it over to Grace Fisher, who will provide a patient perspective.
Thank you. Hi, everyone. My name is Grace Fisher. I'm going to try to get through this without crying, so please bear with me. But I feel the rush of water, the cold as it bites my scalp and numbs my toes. I take a deep breath and dive under a wave, my arms extended, protecting my head and staying under long enough to grab the sand, feel it run through my fingers before jumping up, exhaling and then inhaling the salty air once again. This isn't the act in life that brings me the greatest inner peace, joy, and appreciation of my physical abilities. Swimming in the ocean, dolphining through waves in Southern California. I wonder each time I get this opportunity if it will be my last. If my body will no longer be strong enough to withstand the current. When you live with a disease that eats your muscles relentlessly, you feel an ever-present torture, both bodily and mentally. The pain, the fire you feel in your muscles, and the anxiety of what small or large abilities you may lose, and when. When is the ever-present question. Thank you for the opportunity to speak with you today. I'm honored to be here on behalf of patients and families of the FSHD community. We are desperate for therapies, and we are so grateful for the attention the FDA, scientists, physicians, and pharma companies are devoting on our disease to ensure that we get treatments as soon as possible. I have FSHD. My mother, 2 sisters, my brother, and I all have this disease, and the expression among us is all quite different. I'm actually incredibly grateful for the strength I have because amongst us, I am the least affected. Still, FSHD has greatly affected my life. When you live with a degenerative disease with no treatment and no cure, you live in constant fear. The days of pain and fatigue can be excruciating, but the mental fear and frustration is often as bad as the pain you feel. As you sit there thinking of how your muscles are slowly dying, how you are powerless to stop it, and how you don't know if tomorrow you won't be able to brush your own hair, physically do your job, hold your baby, or get up the stairs. Each day you feel the urgency of science, the hope for some therapy that will slow, stop, or restore function. It is the little things we lose that many researchers and doctors have trouble seeing and understanding that precede the big things: swimming, running, walking, even breathing. These are not insignificant. Someone recently made a comment to me that losing one's mind from Alzheimer's or something would be worse than losing muscles. Well, thankfully, I don't know what that is like. I challenge that idea. When you lose your muscles, you are keenly aware of every slight loss of strength and function. With FSHD, many of us lose abilities at the prime and height of our lives, or even before, in the cases of kids and babies who experience early onset, who are never able to play a childhood game such as tag. This is so unlike Alzheimer's, which generally impacts life after retirement, after you've lived a full life, had a career, raised a family. With FSHD, some of those in our community never are able to play sports, go on hikes, swim in the ocean, never are able to hold our babies close, or even physically carry a child. Imagine what it is like to lose the ability to walk at the height of career advancement, to lose the ability to pick up a pot or chop vegetables when you just need to feed your kids. The timing of this loss for many of us means that we can't live life to its fullest, or that we struggle immensely to do so. The world is not built for the disabled. Without treatment, we are left to rely on inadequate orthotic devices which stab our ribs and are awkward and uncomfortable. Being keenly aware of this gradual loss of strength is juxtaposed with our body's ability to compensate in vast ways. This again challenges the scientific community's ability to understand FSHD. In my professional experience, I've had the benefit of working in the medical and scientific field. I work on the business side of clinical research, specifically that of imaging. As a patient, I have participated in the natural history studies for FSHD. I have performed the functional assessments and have been screened for some of the drug studies. I have not made it into any as I was deemed too strong. And my siblings, who are all variously affected, have participated in both natural history and drug studies. But one was recently rejected as she is now too weak. With my experience in clinical research, I understand how important it is to show significant change during the clinical trial, an incredibly difficult thing to do in a very unorganized and heterogeneous disease like FSHD, and one where the pace of muscle loss varies so greatly amongst the patient community and throughout our lives. This means that finding the ideal participant means finding someone that is weak enough to show the disease burden, but is strong enough to perform functional assessments. Not every patient may be represented in the research, but we all need treatment. As someone who is too strong with this disease, I would like to offer some insight. I can perform the functional assessments pretty darn well. I can climb 3 stairs, I can walk, I can even run. I can squeeze a hand, generally lift my legs, and point my toes, at least for now. This is not to say that I'm performing these functions as I did 5 years ago, 10 years ago, or certainly when I was an aspiring child athlete. Assessments show if I can raise my arms, but not how long I can hold them up before they collapse in fatigue. The assessments show my ability for the one test, but they aren't able to show the fatigue I feel on a daily basis or measure the duration of pain and fatigue after the day of testing. My performance on the tests may also vary greatly by how I felt when I started the day. These tests measure our body's ability to perform these functions, but not how. They can't measure, for example, that when I sit up from a lying position, I have to roll over to my side and use my arms to push myself up. You see, my core is my weakest area and I cannot do a sit-up. Or how if we walk, we may need corrective devices such as a back brace, ankle-foot orthotic, canes, and walkers to support us as we go. When researchers are trying to measure my tibialis anterior strength as an example, I feel my quadricep muscles also activate. This is part of our compensation in FSHD, but probably also how all human musculoskeletal systems use so many different muscles to perform a single function. It's very difficult to isolate a single muscle. With the irregular patterns of disease, as well as compensatory abilities of the human body, the functional assessments are imperfect. Questions may arise in the data. Please look to the patients for insights. We would be more than happy to help. Take one of my siblings as another example. Before she became fully disabled, she would use the handrail for the stair climb test. She would pull, then push herself up each stair to help compensate for her leg weakness. She could climb the stairs, so she was able to perform the assessment, but this test didn't show how. And it certainly didn't show the pain and effort it took to get up the stairs that were both physically and mentally excruciating. It couldn't show that she couldn't get out of bed for days after the trial day and experience weeks of fatigue afterwards. Now at 40, she struggles just to stand, let alone walk. FSHD has robbed her of this basic ability and many others that so many of us take for granted. It breaks my heart as I watch how difficult it is for her to do simple things, peer into the pot on the stove because it's so difficult for her to stand up from her wheelchair, or how she can't climb the 2 stairs in my backyard to watch her son play with his cousin. Every day is an incredible struggle. The subtleties of how we compensate for each weakness tell a story, one that potentially foreshadows an inevitable complete failure of functional ability perhaps, but also of the heterogeneity of the disease, the manifestation within a single patient and among the population as a whole. This is why when I first saw the whole body MRI images of patients affected by FSHD, my eyes lit up. Knowing radiology, seeing the muscles in black and white, how some are so shrunken and some are nice and black and robust, but some are marbled, really spoke to me. Seeing how some patients have robust looking muscles on one part of a limb, but not the other, was like for the first time feeling truly heard. The technology visualizes exactly what we try to explain to doctors about our bodies so perfectly. The name of this disease does not match the many patients' manifestations and does not explain how every muscle in the body can and is affected in some patients with our disease. The MRI images very much demonstrate this. I did want to speak to biopsies. This is a patient population that has been told our whole lives that any muscle we lose, we will never get back. Each muscle is so precious. While we are so desperate for treatments and we will largely do anything to help the science, taking chunks of our precious muscles is not something we take lightly. It would be ideal to find alternative options. Because of this and the nuance and heterogeneity of the expression of FSHD, I wanted to underscore the need to think creatively about how sponsors demonstrate efficacy. The functional performance is nuanced. A one-size-fits-all approach cannot work, and we must evaluate response on the patient level. Last, I want to implore the FDA to honor the fact that without any treatment, the patient community must live in mental and physical anguish as each day passes. We are so desperate to extend the window of our functional abilities, to give us the time to live our lives to the fullest, to play a game of tag, to have a career, to hold our babies, and to swim in the ocean, that even a treatment that might slow progression, as have recently been approved in Alzheimer's, would be a win. We understand that stopping the genetic cause may only be the beginning. We understand that it may take time and additional therapies to rebuild all that we have lost. We can't wait to find out. Please treat FSHD with urgency. We are desperate. Each day matters. Thank you.
Thank you, Grace. Now we're going to have Jeff's up. I'm trying to bring up the slides. Okay, Jeff, please introduce yourself.
Yeah, hi. So my name's Jeff Statlin. I'm a professor of neurology at the University of Kansas Medical Center. I first want to thank Grace for sharing your personal experience of FSHD. I thought that was very effective. Going to switch gears now a little and talk about some of the clinical outcome measure development that we've had going on. This is really very much a collaborative effort. So it represents an international collaboration between academic centers and a collaboration with our industry partners as well. Publication of the genetic model for FSHD in 2010 identified DUX4 as a target for therapy. This led to a translational shift in the field from what causes FSHD to how we can treat it. Now we're really seeing the fruits of what's been many, many years of effort as we see our first molecularly designed and gene-targeted therapies entering clinical trials. Next slide. These are my disclosures. One more. Excellent. When we think about clinical outcome measure development, I think it's very important to put this into the context of what we know about FSHD and the patient experience. And we've been hearing about some of this. When we think about FSHD, it's caused by a toxic gain of function due to derepression of DUX4, which has sporadic expression. So this is really unlike some of the other dystrophies like Duchenne muscular dystrophy, where there's an absence of a structural protein which will be ubiquitously present. This sporadic expression really almost has its correlate in what we see clinically, where we can see a disease that affects people from birth through old age, considerable variability in the severity and the rates of progression between individuals, within a single family from generation to generation, even within an individual from one side of their body to the other. But on average, what we have is a relentlessly progressive disorder where about 20% of people over 50 may require the use of a wheelchair or lose their ability to maintain a job. We see a loss of motor strength or function on average about 2 to 4% per year. There are clinically significant extramuscular manifestations that affect the eyes or ears, but in an average adult population, this is going to be a very rare event, maybe 1%. So very challenging to think of those as an outcome measure. There are no current FDA-approved therapies, and treatment is our multidisciplinary care clinics. And while standard of care may be uniform across the US, it may differ from country to country. Next slide. And when we think about the patient experience, and you heard some of this when James was talking, we've had several very large surveys in the US and also in the EU where we've asked patients about the prevalence and impact on their lives of different symptom categories. On the right, I'm showing the results of a recent survey in the EU of over 1,200 individuals with FSHD. The boxes represent the most prevalent and impactful symptom categories. And if you look at them, they're really almost all related to weakness, difficulty doing things like lifting the arms, mobility, results of weakness, things like fatigue or endurance. And when you look at the future concerns below that, you'll see these are also related to weakness, losing mobility or the ability to walk, or losing independence. So when we're thinking about drugs that improve strength or function would be important to patients, and our outcome measures need to assess strength and function. This is an overview showing some of the current outcome measures that are being used in clinical trials. And we're going to go into more detail about these, so I'm not going to spend a lot of time here, but a few things I just want to point out. We do have outcome measures that represent globally what's going on across the whole body. These would be outcomes like an FSHD functional composite. Our strength measures can be used regionally or as well some to give you a whole body average of what's going on. We have some regional assessments. You heard about the reachable workspace. This is how far you can reach in the upper extremity. We perhaps have a need for more outcomes measuring things around the shoulders and arms, but we have a large number of functional motor tasks related to mobility. Many of these have been used and used as primary measures in other diseases. These are things like the 6-minute walk test, and we have a fairly large amount of data about this for trial planning in FSHD. In terms of patient-reported outcomes, there are two that are disease-specific, the FSHD Health Index and the FSHD RODS, and I'll talk a little bit more about them. But we've also obtained data on several smaller or narrower patient-reported instruments, things like the Upper Extremity Functional Index, the Facial Disability Index, and most of the studies now have included some standard anchoring questions, things like a patient global impression of change or severity. There is a model, though, that's developing for our disease-targeted therapies in terms of what outcomes are being included in a primary fashion, depending on the phase of development. So when we're thinking about disease-targeted therapies and we're early in development, the outcomes that are really sort of taking the front are going to be biomarkers, things like DUX4-related biomarkers in muscle tissue, or MRI and fat fraction as an early signal for what's going on in the muscle. As we get later in development and start to head towards drug registration, it broadens and we start to include those functional outcomes. This can be things like the reachable workspace or strength testing. Can we go on to the next? Yep. There are several studies that are going on now or have been recently completed that can serve as sources of data as we think about outcome measure development. And I want to point out we have several large efforts towards trial preparedness going on the academic side at the same time as we have the industry studies, and that creates a nice synergy between the two so we can learn from what's going on in the clinical trials now and adapt to it in our natural history studies. I just want to point out a couple of the natural history studies because we'll talk a little bit about them and show some data. The Resolve Natural History Study was meant to be a trial preparedness study. It has several measures of strength and function, as well as the collection of biological fluids for biomarker development. It included 320 individuals who were followed for 24 months at 14 sites in the US, the UK, and Europe. The follow-up to Resolve was called MOVE. This is a study where individuals from Resolve could roll into it, but it also broadened the inclusion criteria to include people who were not originally included in Resolve. This could be children or people who were no longer able to walk. Currently, there's 400 individuals involved. We've been following them for just over 3 years at 18 centers in multiple countries. It also has a variety of strength and functional outcomes that overlap with Resolve, so we can have that continued longer time frame. There's sub-studies running on the MOVE study that allow us to add additional measures as needed. As we learn from the experience in the industry-sponsored clinical trials. In this case, we added whole body MRI. We added the muscle biopsy because we realized we needed to know more, the reachable workspace. And in 100 of those individuals, we're collecting a digital motion analysis outcome that's become quite useful in Duchenne muscular dystrophy that includes the stride velocity 95% upper confidence limit. You can see a number of the clinical trials listed here. The Acceleron and Fulcrum programs are now completed. They were a phase 1-2 study for Acceleron and phase 1-3 studies for Fulcrum. You can see the numbers who were included that ranged from 80 to 90 up to 260 in our first phase 3 study in FSHD. The REACH study really was a proof of concept that we could do a phase 3 study in FSHD, enroll in it, and get it done in a reasonable time frame. The primary outcome for the phase 3 study was the reachable workspace, which we'll talk about, which unfortunately wasn't able to show us a treatment benefit in that study. The model I mentioned for study design from phase 2 to phase 3 was really developed in these programs with use of biomarkers and MRI in those phase 1 to 2 studies, which then transitions into our functional outcomes for the later phase studies. Next slide. This is just an illustration of our current trial landscape. I want you just to center on those blue bars in the middle. These are our molecularly designed therapies, RNA therapeutics, true gene-delivered therapies. And you can see the two that are currently in human studies. But I think what I really want to pay attention to are those 5 to 8 studies that look like they'll be going into their first in human study in the next 1 to 2 years, which makes this a very important time for the field. Next slide. I want to start by talking about strength. This was one of the first outcomes developed in a very systematic way for FSHD. And as you heard, it's a very important concept for patients as well. There's 2 widely used methods, manual muscle testing and quantitative myometry. You can see them illustrated on the right on this slide. For both, you can use standardized positions to obtain that strength and could look at individual muscles or combine them for a whole body composite. There was a natural history study done in the late 90s that really helped develop these outcomes. They showed good to excellent inter and intra-rater reliability in a multi-center setting. And strength was the most sensitive measure to change in that late 90s natural history study with an effect size of around 0.3, which would be approximately 2 to 5% loss of strength per year. Our subsequent Resolve and MOVE studies have really validated these findings, seeing very similar rates of change from year to year and very similar variability in those measurements. This allows us to make fairly particular sample size estimates. So if a drug was just going to halt progression in FSHD, you would need about 160 per group. However, if a drug halted progression and then these people improved by a similar amount, suddenly the number needed is actually quite reasonable for a clinical trial, 40 per group. Can we go to the next slide? The FSHD functional composite was developed by the group in the University of Rochester. This was a response to those early clinical trials in FSHD, the feeling being that we needed a functional motor outcome that really represented all areas that were affected in FSHD. It was based on some early surveys of patients where we took each of the functional categories that people identified as being prevalent and impactful, and the evaluators chose already validated and well-established functional motor tasks to represent each of those areas. There's 18 separate items included. It takes about a half hour to complete. Several of the items have already been used as primary outcomes in other muscular dystrophy trials, things like the 6-minute walk test, the ascending and descending stairs. It shows excellent retest reliability in a multi-center setting with an ICC greater than 0.9. Factor analysis supports this 4-factor solution, so it really supports the breakdown into these 4 basic functional categories. Using a statistical technique to determine the minimally detectable change that would be required to be confident you're seeing movement. This would be about a 10% change. It shows moderate to strong cross-sectional associations to other things of interest in FSHD, disease duration, strength, patient-reported impact, and we see a change of about 10% at 2 years. The reachable workspace, this was really designed to fill a gap in the field. We really had no quantitative measure of shoulder and arm function. It was developed by a group in California and then adopted by Fulcrum for their drug development program. It's a novel repurposing of an existing technology using a stereographic Kinect camera to calculate the volume of space that someone can reach into. If you look on the top right, you can see the 4 quadrants around the individual. There is a 5th quadrant, which is reaching back and behind the individual. Also shows excellent reliability in a multi-center setting with standardized training with an ICC greater than 0.95. You can see good correlations between measurements of reachable workspace and upper extremity strength measurements, as well as the ability to do motor tasks. On the bottom right, you can see that there's differences in the reachable workspace area based on your ability to do things like take a shirt on and off, or pick up clothes. In the Resolve natural history study, however, in adults, it's fairly static over time, seeing about a 1 to 2% change over 1 to 2 years. And in the clinical trials to date, in the phase 2 study, there was an improvement in reachable workspace that was seen, but unfortunately not replicated in the phase 3 study, where both groups improved slightly over the course of 1 year. Can we go to the next slide? In terms of patient-reported outcomes, there's 2 I want to highlight. The health index, this was developed using FDA guidance for industry for PROs. It started with individual interviews with people with FSHD. The symptoms they brought up were coded based on World Health Organization definitions. Then the list of things that people had identified as being important was sent out in a survey to over 300 individuals who ranked them based on their prevalence in their lives and the impact on their lives. You can see the top 14 symptom themes listed here, which become the domains for the item. And again, you can see recapitulated that the top items that patients are identifying are really motor, motion in the shoulders, ability to walk, strength in the core. This results in a 14-domain instrument with 116 items, but the way you rank them is similar across all of them. It's a 6-point Likert scale that ranges from it doesn't affect my life at all to it affects it severely. It takes about 15 to 30 minutes to complete it. Can we go to the next slide? The FSHD rods is a rash-built instrument, so also using standard psychometric approaches. It's a narrower instrument, so really centering on functional activities and participation. You can see the 32 items that ultimately were included in it. There's a fair amount of overlap in these items between the 2 instruments. In this case, the scoring is broken down from ability to do it without difficulty, needing to make adaptations to complete the task, to being unable to do it. And you can see them ranked from being something that would affect people quite early, like losing the ability to run, to something that would be a much later impact on someone's life, the ability to fill out a form. Other instruments that have been used include the NeuroQual and the Patient Global Impression of Change, and we're currently analyzing data from the natural history studies and clinical trials to understand how these respond to therapeutic interventions. So we also have had several studies looking at digital outcome measures. Most of these have been pilot studies or smaller single-site studies. There's several ways you can think about using these digital health outcomes, either to measure functional activities or patient reports of function. Most of them have really been motion analysis. So these are tiny motion sensors that you would attach to your body, or cameras that would capture your motion. These include systems by SysNav, APDM, Biosensix, and Actigraph. We're currently obtaining data in larger data sets. In the MOVE Plus study, we have a study of 100 individuals who will be followed for 2 years. I think there's a lot of interest in this approach and technology, and we'll see more of this as we go forward. Next slide. I do want to take a few minutes and give an example of how we can use data from studies like Resolve and Move to understand our outcome measures and how we'll use them in clinical trials. This example is for the 10-meter walk run. This is the most sensitive functional motor task that we have to disease progression. We've seen it in Resolve. It's been recapitulated in the Move study. You can see it illustrated here. You just ask people to go 10 meters as fast as they can, safely do it. It doesn't take very much time to perform. It's an inexpensive thing to do in a clinical trial. We're able to train people in a multi-center setting, so we get very consistent values from one administration to the next with an ICC of around 0.97. And there's nice cross-sectional associations to other aspects of the disease that would be important, things like disease duration, severity, the strength testing, and the patient-reported disease impact. On the right, you can see individual muscles from the waist down out to the distal lower extremity and their relationship to performance on this test. You can see that the key muscles driving that performance are hip extensors and knee flexors, and these obviously are important for an acceleration maximal performance task, but are also very commonly affected in FSHD. Can we go to the next slide? We can also use data from the MOVE study to sort of understand how performance on this instrument fits within the context of changes that are occurring in FSHD. A performance of 0 to 4 seconds is what we would see in a normal population. On the right, I'm showing a histogram of performance from the first visit on the MOVE study, and you can see about 20% of people will have a relatively normal performance on this outcome. As you move into the 5 to 10 second range, you're now in a range where people are going to have changes on their performance in the 10-meter walk run that relates to changes in their patient report of what they can and can't do in the community and in their lives. At the point we hit 10 seconds, we start to see hard transitions where people now start to need things like orthotics to help them move around. At 15 seconds, the need for a cane or walker, and at 20 seconds, the need to use a wheelchair to get around. Unlike some of our other dystrophies, there can be a very long transition period as people are going from being completely independent with ambulation to needing to use equipment that can last many years. And finally, when we think about what size of a change is going to be meaningful, we use a variety of different approaches to get at this concept. One way to do it is to look cross-sectionally at how it relates to a patient report of function. We collected something called the current ability score, which just asks people about their ability to do common daily tasks like transitioning from a bed or a chair. Each change of 1 on that item is equivalent to losing a key function. And you can see on the bottom, in those normal ranges on the 10-meter walk run, you're not seeing a lot of movement on that scale, but then you start to see a very much a stepwise increase in the burden on the current abilities based on the performance on this test, with the change of 1 in current abilities roughly equating to a change of 0.9 seconds on the 10-meter walk run. This is also the size of a change we see on an annual basis in a large population of adults who are clinically affected, so a change of about 10%. And this also relates to the size of a change that we would estimate as being important, either using a statistical technique like the minimally detectable change or an anchor, where people are ranking if they're having a little more or a little less trouble over the course of a year. This is just showing our different outcomes and which ones are being used in the different studies that have been either recently completed or ongoing. What I want to point out here is there's a considerable amount of overlap between the natural history studies and the clinical trials. This allows for post hoc analysis to get a better understanding of how these outcomes are performing, but it also allows the clinical trials to compare against a natural history data set so they can better understand what's happening in the placebo groups and the treated groups in the clinical trials. Finally, I want to just finish talking a little bit about how we think of inclusion and exclusion criteria. Are there selection criteria we can use to select for people who are more likely to change? And then finally, thinking about how large a change is meaningful. When we think about selection for clinical trial, there's several factors that have been proposed to potentially impact performance over 1 to 2 years. These include things like genetics, gender, and baseline functional status. Of these items, the only one that really seems to have a clear impact on our ability to select people who are more likely to change is that baseline functional performance. And it turns out this is a theme I think we're going to hear repeated today as we go into the MRI, where you're going to hear something similar in that muscles that are already partially affected but not too affected are most likely to change over a shorter time frame. Can we go to the next slide? This is a slide prepared by one of our industry partners who used our Resolve dataset for their trial planning. And what they did here is they calculated something called the standardized response mean for each of the outcomes. This is just the change at 1 year divided by the variability. A value of around 0.3 would be a small responsive outcome, 0.5 would be moderate, and 0.8 would be a largely responsive outcome measure. You can see the array of outcome measures listed here based on how large a response mean they have. You can look at the quantitative myometry at the top in the box and see that that response or that effect size is around 0.3. This is what I said we had seen in the natural history studies. So it's nice to see that we're recapitulating that here. So getting consistency and doing something very simple by selecting individuals who are in that dynamic range for strength testing. So say between 20 and 80% of predicted, or in the dynamic range on something like the 10-meter walk run in that 4 to 10 second range of performance will get you a much more consistent change over 1 year. And you see those response sizes increase up to that moderate level just by using that selection criteria. Can we go to the next slide? Finally, when we think about meaningful change, there's a few different ways to think about this. One way would be just to look for hard motor events that are occurring in people's lives. Things like losing the ability to walk or needing a wheelchair. And indeed, we have data about this, and you can calculate the rough annualized frequency of this event occurring, which is about 2% per year. But this is a very low frequency, and it would be very hard to design a clinical trial around looking at some hard motor event occurring during the conduct of that clinical trial. So short of that, we have to think about other ways to figure out how large a change would be meaningful on our outcome measures. Can we go to the next slide? And in order to do that in our trials, we use a variety of techniques. And what we're going to look for is consistency in the size of that estimate across the different approaches that we're taking. We can use a statistical approach. This would be something like the minimal detectable change that you're confident a true change is occurring. You can use an anchor where you ask people, are you feeling a little better, a little worse, a lot better, a lot worse? And you can see the size of a change that corresponds with someone saying they feel a little better. You can look at the cross-sectional relationship to people talking about what kinds of functions they can and can't do. You could look at what is the change we see over time in individuals with the disease. And finally, if you have the data, you can actually model what performance today on these outcome measures will predict that hard motor event over the next 1 to 2 years. This might be the need for a wheelchair or other assistive mobility device. In general, our estimates have been fairly consistent across our strength and functional measures, being about a 10% change being meaningful. So as we go towards the future, we have several natural history studies that are ongoing that are working synergistically, I hope, with our clinical trials. And as we learn something new in the clinical trials, we can take it back to what we know in those natural history studies. Biological fluids and samples are being collected to allow for further biomarker development with a shorter time frame, because we'll have already well-described clinical cohorts to compare against. And then finally, we really need to be doing the same kind of work we did in adults in children. And I'm really pleased to say we do have NIH support for a study in pediatric children with FSHD that should be starting sometime this month. All right, thank you so much. I'll go back for the discussion.
Thank you, Dr. Statlin. So now we have a number of discussants who work in this area with outcome assessments, who are poised to engage us in some questions. And I'll start with Dr. Hayward, Dr. Larry Hayward from UMass Medical School, Chan. So we just heard Drs. Melian and Statlin describe the phenotypic variability in individuals living with FSHD. How, in your opinion, might a clinical trial plan manage this variability as they work to assess muscle function?
Yes, hello everyone. I'm Larry Hayward at UMass Chan, and I think as Dr. Statlin emphasized, the clinical heterogeneity in this condition is extreme, and I think some of the points he made I would like to reiterate in terms of the focus on motor function, which in many ways relates back to the patient concerns, whether it be ability to do activities or having an impact on their concern and anxiety about the future due to uncertainty of their progression of motor function. And so what can be challenging is that, as Dr. Statlin emphasized, the power of a clinical trial depends on identifying a change that's meaningful in a trial. And he mentioned both global measures and more specific measures. And I think the power of these natural history studies is that there are ways to sort of link some of those together. And there are certain measures that are coming out that are relatively simple to perform that may offer more informative change over time. At the same time, I think there are patients that maybe don't fit into the usual Ricci progression, and there may be baseline characteristics that we can identify by MRI that might then help to dictate which subgroup of FSHD phenotype might be worth following more closely with certain outcomes that are tailored to that disability. And then over time might prove to be more sensitive, at least for looking at that subset of patients. At the same time, obviously, one wants a treatment to be validated to be shown to be useful for patients as a whole. And so there's a conflict between those. But I think that it's exciting to me that there's so many measures now that are becoming more granular. So we're obviously, we'll be hearing more about structural granularity and whole body MRI. But also in terms of functional tasks, we're starting to try to think about measures that might go beyond a simple time measurement for a task to actually looking at how the combined effect of different muscles and how you can break that down into perhaps individual muscle function. And that's important because we want to capture not only weakness and inability to do a task, but how a patient's other muscles might be compensating. And that can feed into other concerns of the patients, such as the development of pain due to overworked muscles that are still relatively unimpaired. So I'll open it up for if anyone else has some other comments.
Thank you, Larry. I'd actually like to go to a different question with Dr. Jay Han. Jay, so you and your group were involved in the development of reachable workspace, as Jeff described, to start to address the upper extremity challenges of an FSHD subject. So what remains to be done with reachable workspace and other upper extremity endpoints that we have in our hands?
Thank you very much for FSHD Society and also Jeff and Larry, and everyone else, and Grace for sharing the patient voice. I think the field has moved tremendously, and we know so much more about the outcome measures that we did before. Yet much remains as to the clinical meaningfulness of these outcome measures. In a small way, that we're trying to collect and trying to extrapolate what it really means for the patients. So that was really the motivation for our group in the past, that not just to limit to strength, but really, what does that mean? And reachability as an outcome measure for upper extremity is important. We use our upper extremity to interact with the world, grab something, bring something to our body, bring something to our mouth, brush our hair, put on clothes, in various different things. So we felt that reachability is an important outcome measure to look at. Certainly, it's not all-encompassing, and it doesn't take into account a lot of different factors, but that was one of the motivations that we worked on. As patients have said in the past, and I think one of the patient's voice mentions, it's like FSHD, your arms being tied down with rope, and you are unable to move. And your independence, activities of daily living, the basic things that we do, and really tied to quality of life is the mobility of your upper extremity. So the strength measures and the reachable workspace and other outcome measures that we're working on really speaks to that. Yet the connection between the strength and what it really means in a day-to-day activity is really lacking. So imparting more meaning to some of these strength changes and also to impart meaning to reachable workspace, I think is a necessary element that we still need to work on. The other thing is mobility. And as Jeff mentioned, there are lots of outcome measures that are coming on. There's looking very promising. But as a physical medicine and rehab specialist in the neuromuscular realm, the transfer is really the basis for mobility. Before you stand up and walk, really, can you get out of bed? Can you move from lying to sitting position? Can you move from sitting to standing position? That's really elemental and sort of the beginning part. And I know some of the patients voiced really a difficulty in those transfer activities. So I just want to kind of put in some plug there for us to, as a outcomes researcher myself in this area, that's an area that hasn't really been looked at as much because we've looked at, say, upper extremity and lower extremity and ambulation, but the transfer activities is elemental for all activities. And it really gets up to the truncal, the torso movement and weakness that Grace mentioned also. So I'll stop there. Thank you.
Thank you, Jay. I want to talk with Tina Duong about statistical approaches to meaningful change can be challenging. So how have exit interviews and exit surveys been used in FSHD to evaluate meaningful change?
Thank you so much, Lucian. So I'm actually, my clinical training is in physical therapy. So I think a lot of this has to do not only with the statistical approaches, but the psychometrics of some of these outcomes. Jeff did a wonderful job displaying all the tremendous amount of work that has been done in the last 2 decades to get us there. I think whenever we're looking at minimal detectable change and minimal clinical important difference, we have to think about it not broadly, but it has to be tailored and specific to not only disease, but the stage of the disease and the function. As Jeff stated earlier, the baseline characteristics are very predictive of what these MDCs may be. Some of the things that we can increase sensitivity is one, standardization on how things are done so that we can decrease the variability in our methodology, whether it's strength testing or functional testing. And that has been really well done with the collaboration between some of the academic natural history studies and the drug developing partners. One of the other things that we need to take a look at is to increase sensitivity, not only is by the stage of the disease, but maybe the rate of change. There's a lot of talk about we can learn a lot from the examples of SMA and Duchenne, in which, particularly in Duchenne, the latest approval from the FDA was looking at rate of change. So you're not looking at just the mean difference between baseline and 12 months, but you're taking into consideration the rate of change and how does that rate of change anchor to a clinical important difference. And as many of you can imagine, clinical meaningfulness may vary, and it varies even more for a heterogeneous disease such as FSHD. So then we have to have what's our context of use of this MCID? Are we looking at the change related to quality of life, function, or change in healthcare or standards of care that we would provide? All these will give us different MDCs or MCIDs. And then not only that, but they are going back to the approach. One of the recent things that we looked at for MCID and MDC is that in SMA, there's a scale very similar to the FSHD COM called the Revised Hammersmith Scale. And we looked at it at different cohorts: pediatric, adult, those who can walk, those who cannot walk, and different experience of evaluators. The MDC varied between 2 to 7 points. And so knowing that, what we've done working closely with the academic group is to be able to define that so that we can help some of this statistical modeling and power analysis that may be used. And then lastly, one of the things that I think about a lot is biomechanics. In order for us to increase the sensitivity, we have to decrease the variability. So how can we learn from not only the quantitative muscle testing, why does it or does it not correlate to some of the function? Quantitative muscle testing that is typically done, or MRC, is done in an open chain fashion. What that means is that your hands or feet are not planted to the ground. Functional tasks are related to closed chain activity, so you have to activate your muscles differently. Therefore, in FSHD, when you have different parts of your muscle being affected, that may affect how you function. The other part is, as Jay was mentioning, the trunkal deviations. Because of the abdominal weakness as well as the trunk extensor weakness, we've seen as physical therapists when we're evaluating patients, if they even have slight movements of their trunk, that's going to affect their ability to lift their arm. And it's very, very small. Therefore, some of the technology that we are working on is understanding better some of these explanatory variables that may be better assessed by motion analysis or kinematics to explain some of those differences. And I think that explanation will really result in improved sensitivity, therefore better reflect some of these clinical meaningfulness in our approaches.
Thank you, Tina. That was great. I'd like to go to Dr. Johanna Hamel now to talk about electronic PRO measurements. So can you speak to the considerations given to electronic PRO measurement approaches in FSHD, and is this currently being evaluated to complement the performance measurements?
Yeah, thank you for having me. And as you know, I do have experience with FSHD patients in the clinic, mostly recruiting for clinical trials, but also natural history studies, as well as some experience in other muscular dystrophies like myotonic dystrophy. And so I think, obviously, I don't have to highlight into this audience that PROs are extremely important and are, key in evaluating and really getting at that meaningful change, evaluating patients. I think there are a couple things that I want to say up front, because the PROs are only as good as we design them. And so, the questions are often not as participants would have phrased them, or if it was up to them, they would have rephrased them, even though, researchers have taken the best effort to really have the patient input in development, but they're never perfect. And so taking that into consideration up front, that they have their limitations and they don't capture the full picture. I think they're extremely helpful. I think the one thing that is always important to say, well, first of all, people are capable of filling them out. They will fill them out. It's important to make it as easy as possible. That's the feedback I get from participants. Whether it's on an iPad or a device, there are often issues with those technical issues. If that can cause frustration. To make it as easy as possible and not deploy them too frequently so that it gets kind of an automatism to answer them. There's an art to that, and I think about that quite a bit. How should it be displayed? What should it look like? Because if you move from paper to electronics, that's important. Should there be one question per page or not? Things like that. That's my take. And then, so for example, if you deploy them daily, and sometimes we get greedy, we want more data, that can be a risk because then there's a fatigue in filling those out. And some things, particularly if there are symptoms that really don't change much from day to day, you don't want to ask that question over and over. So I think it's extremely important to have the patients part of that process and think about it. What would it look like to fill that out on their end and how frequent? Now, one thing I want to highlight in terms of patient-reported outcomes, because in my experience, what has been incredibly helpful is actually the conversation in a trial. So the PI participant conversation at each visit, asking how things are going, really exploring it, not with just one question and a PRO, but really with a follow-up question and really getting to the bottom of an experience. I think that's important. I know this is not well captured, maybe in those, global impression of change, either patient filled completed or clinician completed, which I think are important. But other than that, a lot of that gets lost. So that goes back to the exit interview that you were mentioning earlier, which can be extremely important, in my opinion, to capture some of those things, because we will only capture what we ask about. But a lot of things are happening that we didn't know about, that we may not know that they're FSHD, that we may not know that they're responding to treatment. And in these early trials, I think it's important to keep that in mind, the limitations where we are in research, and to be open to learn with each participant as we go.
Thank you so much, Dr. Hamill. I'd like to go back to Dr. Michelle Mellion to comment on, we've seen trends moving in clinical evaluations of novel therapeutics that may not necessarily meet a statistical endpoint. And I wonder how you might recommend in FSHD, we think about these things as clinicians.
Yeah, thank you, Lucienne. And I think that Joanna has touched on this a bit, as well as Jeff and Grace in her beautiful narrative about her personal experience with FSHD. It's really, as people have said, a heterogeneous disease. And so the way that people experience FSHD and where they are might be different. And it's difficult to stage this disease specifically to fit people in specific stages. Although we do have, Jeff has presented some very nice data to show where those milestones may be. One of the things that we as a community may want to consider as we're looking at these myriad of endpoints and the data that we've developed is thinking about looking at these endpoints in terms of trends and not necessarily statistical significance. As we know, it can be difficult to find that perfect patient, which Grace had alluded to. And so in order to understand the potential impact of these new and innovative therapies, we may have to consider as a community, as well as looking at statistical significant assessments on endpoints, evaluating the trends on different markers that may be important to patients. And Joanna did touch on this a little bit as well in trying to identify what is important to patients and how is the disease affecting them and your patient experience, and then following up on that over time within the clinical trial, which may essentially be more meaningful in the end in terms of even finding MCID or stat sig on another endpoint that may not necessarily be as meaningful for patients. So I think as we as a community move forward in designing these clinical trials, and I know that many of my colleagues on this call are already doing that, is to not forget to include that patient experience and also learning how to quantify, at least by trends, to understand what the impact of potential treatments may be on patients from these treatments.
Thank you, Michelle. And I think I'd like to go back to Dr. Statlin to kind of wrap up this portion with a question of, Jeff, if you think if a therapeutic benefit is seen in a clinical trial population, is it reasonable to extrapolate to a more diverse FSHD population who didn't meet the inclusion criteria? Would they benefit?
Yeah, I mean, that's a great question. It comes up a lot. We're seeing it in some of our other neuromuscular disorders where new therapies have been approved, and then you see efforts to try to limit who can get it to the population who was included in a trial. But I think the whole concept and the infrastructure of these experiments to gain evidence to support, marketing of drugs always has the assumption that we're going to do an experiment in a more limited group. And, if our assumptions are right, it should generalize across the broader population. And I think if you've got a disease-targeted drug and it's, you think it's going to hit its target and modify this disease, you may look at some ambulatory measure, but the question would be if someone's in a wheelchair, would this benefit them? The answer would be yes. It'll benefit them to the degree that they have function to preserve and save and, benefit from that treatment. And so we need to remember this as we go forward, that, the trials are always meant to support this broader generalization as you go to the market. And part of the phase 3 program is to maybe try to get some information about these other individuals, but then sometimes you gain that information once it's on the market and you continue to follow people in registries so you can really understand who's benefiting and maybe who isn't.
Thank you.
Yeah.
Thanks to everyone who was presenting and involved in that discussion. It was really beneficial to learn the exceptional amount of work that has been going on with selection of outcome assessments and the use of PROs. So we're now going to move on to the next portion of our discussion, which is on MRI. And Dr. Seth Friedman is going to talk us through when the field first started thinking about how to use MRI in FSHD. He was the first source that the field went to in really thinking about how MRI can be applied. Seth?
Thank you, Lucienne. So, hello, my name is Seth Friedman. I am at Seattle Children's, and you'll have to excuse me. It's very sunny here in Seattle, and so none of us really know what to do when there's sun. So what I'm going to try and do today is give kind of an overview of how the field has progressed over the last almost 20 years. And I really appreciate all the talks and the commentary before this. I hope it'll give some good idea of sort of how our thinking has changed as well. And at the end of my slides, I'll kind of give at least maybe a little foreshadowing of where we're thinking we can go analytically now with the field. So really in FSHD, there are, the main contrast is very similar to what has been used in Duchenne's with his Dixon imaging, which gives you 2 image contrasts of water and fat. There's also been a lot of interest in imaging of inflammation with short tau inversion recovery imaging, or STIR. And there have been many other contrasts that have tried. We've certainly done many other different types of quantification, and these are interesting for looking at things like, is there fibrosis, is there diffusion, but they really haven't been very useful biomarkers to date. And next slide, please. And so the first phase of the field was really qualitative, trying to measure images using rating scales similar to what you heard about in some of the functional measures. This was an early study by Doris Lung in Johns Hopkins, where they studied 14 individuals across the whole body. And what you can see here is that yellow is a normal appearing muscle and red is a very fat involved muscle. And what you can notice across these 13 subjects is that there are really a lot of involvement across the entire body. And the other thing that's notable is that there's a lot of sort of twinkly distribution, which is there's not a lot of symmetry in many muscles. And so this was sort of one of the first illustrations that it's a whole body disease. And the advantage of this is you can obviously rate all the muscles in the body. The disadvantage being it requires an expert and it's very insensitive to seeing change over time. Next slide, please. As we've moved on to quantitative techniques, these are really advantageous in that you can get more measures of different components like fat and lean muscle. You can objectively track patients over time. But it was very, very difficult until recent years to measure the entire muscle from its proximal to distal extent. And so the other challenge is that even if, as you start to think about this in a computer vision standpoint, it can be very difficult to train the computer to discern muscles that are fatty and to discern muscles when they all converge towards the tips. And so you'll hear some more about with Sylvia's presentation how new AI methods have really been incredibly helpful in solving these problems. I also see there's an AI question in the chat, so we'll get to that. Next slide, please. And so as the work has progressed, really what we've been highly focused on is trying to get as much explanatory variance out of the MRI as possible and sort of removing any error variance. And so this is a paper from Lara Ream and colleagues in Scientific Reports. And really, we can now derive from an MRI, we can derive a whole range of volume metrics, things like lean muscle, fat volume, and also bone volume, which we'll talk a little bit more about. That's very important in kind of constraining how the progression occurs. You can also convert this into what these cross-sectional metrics where we plot the muscle from the proximal to the distal extent. And on the top, you can see things like the red solid line is the muscle. A year later, you can see that the muscle decreases. Fat is shown in yellow, and you can see that that is more prevalent at one end than the other. You can make a fat infiltration map, which this fat fraction map is primarily the thing that we talk a lot about in FSHD. You can also start to make these pixel visualizations, and while these seem like incredibly visually very novel, they've also told us some very important things about how muscles progress. And so, for example, you can imagine in this visualization here, if you were to select just a central slice, you will not really represent the fat pixels, which are shown in yellow. And so being able to analyze the whole muscle is very, very important. Next slide. So we've also learned some just things that have been, I think, they seem obvious now, but they're very different to how we conceptualize the disease. So for years, people thought the disease kind of fills up with fat like a cup, like it starts from the bottom of the muscle, and then it goes from the distal all the way up to the proximal side. And these are some plots of different muscles, and you can see, I'll just sort of walk you down the patterns on the left. So some muscles really have sort of a homogeneous fat distribution that progresses across the whole muscle. Some do start from the distal end and fill up. Others start from the other side. They go from proximal down. Some start from the ends and fill towards the middle. And then others start at the middle and proceed outward. And this is not necessarily true. Like, so for example, a hamstring muscle doesn't always have the bottom pattern. So there can be a lot of variability within muscles of how they change. Next slide. So these pixel maps were also really, really, I think, super helpful for me as someone that's tried to analyze images over almost 20 years, that you can really see how the disease changes over time. So this is an individual patient, and you can see change up in the glute muscles and in the lateral gastroc. And so this is very affirming of what we see mathematically, where we would say, okay, the change goes from 10% to 20%, and this can now be visualized. Next slide. And so the challenges you've heard about, and I think that we're oftentimes struggling with, is that this is not a homogeneous presentation. And so on the left, you have two different patients at baseline showing up for a natural history study. And 1 year later, you can see that there are different colors represented. On the left, you have fat infiltration at baseline. And on the right, you have change in fat infiltration over 12 months. And you can see that the patient at the top has a very dark green region in their hamstring. And the patient in the bottom has a very dark green region in the lateral gastroc and a range of other sort of light green in the thighs and in some of the sort of lateral musculature. And so this is sort of the example that we've always sort of struggled with, which is these two subjects will probably not have the same functional decline because they're having different muscles involved. Next slide. And so what do we know? So from cross-sectional findings, I think it's very, very clear now that this is a whole body disease. And Sylvia will show some sort of the exhaustive quantitative data that we can now get from these subjects. There's also very high, variability is the rule, not the exception in FSHD. So patients have a lot of variability between. I think that was also a really lovely illustration from Grace, from her siblings, that all have very different expression. There's also a lot of difference across muscles. So a muscle like the semimembranosus in the hamstring may have a lot more disease than something like the tibialis posterior, which typically is involved later or lesser in the disease. And then we can also see all these different patterns within muscles. Asymmetry is very common. And even though a lot of muscles have fat infiltration, what we kind of consider to be the phenotype of the disease, this confluent blobby fat, only about 5% of them will have what looks like inflammation on stir. Next slide. So what do we know across time? So across time, many, many studies now have looked at like a composite body or a bulk muscles. In that way, in the same way, when you look at the composite function, you see a relatively small amount of change per year. So on a human scale, it's a couple percent at best. When you look at individual muscles, they definitely progress at much faster rates, with some having, sort of what we might consider an escalated course. But some of the modeling work that Sylvia will present, we really learn now that this is specific to muscle identity. So if you have two muscles that both have a 20% fat fraction at baseline, the muscle type and the muscle identity will make a big difference in how they change. The pattern of fat also makes a difference. So in some of the new predictive work we've done, really, the pattern is one of the best predictors of how change will occur over a 1-year period. And then factors like bone volume are very important, and this will be extremely important for upcoming pediatric trials. And on the right-hand side, you see this is from Ian Woodcock in Australia's data, where you have a child over a 14-month period. And this is the same child. These are scaled to their body size. And you can see that the kid's just getting bigger. And so we really do need new ways. And bone volume is a good starting point to characterize normative growth from FSHD progression as we move into these pediatric trials. And the natural history study that Jeff mentioned that we're starting to do will really provide a lot of data on that. And we also don't see stir change much. The one last thing, we did an initial pilot study with tacrolimus and with steroids, and we were not able to modulate stir. And so stir still is one of those things that we're still interested in inflammation. I think it's being looked at in a lot of trials, but we're still not entirely clear how to conceptualize it in the progression course. Next slide. And so very briefly, I just want to talk about, I'm almost out of time, so I'll kind of zip through this. So cross-sectionally, the key finding is that more fat is worse function. And the problem with this is that, as Larry mentioned, and I think Jeff mentioned, and Grace, is that most subjects will not have a consistent muscles at baseline that are affected. And so what this really translates to is that if a patient shows up and they have that transitional shoulder musculature, the best kind of reporter will probably be a shoulder task for that subject. And if a different subject shows up and they have leg weakness, it's possible that a walking task may be a better reporter. So we really have not yet kind of put together the MRI data with the functional outcome data to try and make a slightly more personalized reporting equation. And so I think in the instance of time, if you go to next slide, Lucienne, and then I just will, I'll just very briefly. So, you know, as been discussed, oftentimes we need an approach that really takes into account the heterogeneity of the MRI signal and the heterogeneity of the functional signal. And so if we just simply use these composites alone, these composites may take very large numbers of subjects and very long trial durations. And we really think that being able to create more weighted composites, or to be able to track the specific muscles and musculature per subject that is affected, and linking those to the task-specific behavior that those muscles observe, may be a much more effective way to decrease your subject number, to create a more efficient trial duration, and ultimately be able to really understand how the disease, how a drug is modulating the disease course. And so, Lucienne, I think we can get to those 2 slides in the questions that come up. Thank you very much.
Okay, thank you, Seth. So I'd like to move on to a case study using the data that was derived from Fulcrum Therapeutics phase 2 study Redux 4, where whole body MRI was applied for the first time in a clinical setting to FSHD patients. It was developed initially by Dr. Diego Cadavid, and then Michelle Melian also continued the extension of the development. But the concept of developing biomarkers for use in multi-site trials to describe the progression of this highly heterogeneous disease was something that he thought, and Fulcrum agreed, was very important for our understanding. So now I would like to introduce Per Wedholm, who is from AMRA, a Swedish company with whom Fulcrum worked to develop the whole body MRI. Per?
Yes, thank you, Lucienne. So yes, I will begin and describe these experiences on designing and applying MRI biomarkers for the Fulcrum's losmapimod program. And the aim of this project was, as Diego's citation says here, that develop MRI biomarkers suitable for use in multi-site clinical trials that objectively can describe the progression of disease before clinically apparent effects are evident. Next slide, please. You go back one.
Sorry if I, yeah.
Yeah, so and I will be briefly discuss the developmental journey of this project with a focus on 3 different areas. That is the regulatory requirements on clinical trial biomarkers, a little bit of how we designed and validated the biomarkers, and also report on its use in clinical trials. Next slide, please. So, since the aim was to develop MRI biomarkers for use in clinical trials, great efforts was spent on understanding the requirements on imaging biomarkers in these settings, both from a regulatory perspective as well as operational aspects, as this is a very, very different environment from the usual use case of such biomarkers in academic research. On a high level, these requirements could be summarized in 2 main areas. Minimize variability in all stages of analysis with standardizations in all aspects like site training, MRI scanners, certifications, standardized scan protocols and settings. AMRA has, before this, also developed proprietary and patented image calibrations techniques to get consistent results between scanners and different imaging protocols. Also have semi-automated segmentation and dual operator quality control from all segmentations at all time points to ensure consistency both over time and between patients, as well as continuous image quality control of incoming data throughout the studies, making sure that all sites are adherent to the protocol, identifying problems, enabling rescans, incorrect time windows, and so forth. And having processes in place to ensure data integrity and traceability are up to task for regulatory review and audits. Next slide, please. And when deciding what and how to measure with MRI, certain disease characteristics had to be addressed, such as the accumulation of intramuscular fat and loss of muscle tissue seen in the disease. Also, the heterogeneity of the disease and the slow progression. And as such, we chose to measure both muscle volume and intramuscular fat, since that has a link both to pathology but also to muscle function. And muscle fat, we chose to measure in two different ways, as it's seen in the top row of the images here, where fat is yellow. And to the left, we have muscle fat fraction, which is basically a measurement of how large portion of a given muscle that consists of fat. And then to the right, we have muscle fat infiltration that assesses how much fat is present in the remaining muscle tissue, excluding regions of the muscles that are totally fat replaced. And on the bottom, we have muscle volume as lean muscle volume, just removing all fat from the muscle. To address the heterogeneity, you can please go to the next slide. To address the heterogeneity of the disease, and at the same time use the fact that muscles with increased muscle fat tend to progress faster than normal appearing muscles. Endpoints was defined as a composite, where muscles with similar disease characteristics are joined together to enrich the analysis of change over time and treatment effects, while at the same time take disease heterogeneity into the consideration. And this is illustrated here with two different individuals with FSHD, and highlighted in yellow are these intermediate affected muscles that are assumed to be the ones that will progress the most in the short term. And those are the muscles that we will include in the composite to track change over time. And by this, we can address the disease heterogeneity of the disease, since each individual gets their own personalized set of muscles to follow. Next slide, please. Yes. And the resulting methodology, quantification performance, as well as correlation to muscle function and more complex tasks such as time function test, was evaluated in a biomarker study. Next slide, please. And the first part describing the methodology in detail, as well as the technical performance, was presented in several scientific conferences and later also published in 2022. And next slide. And in the same way as the technical performance and methodology, the ability of using MRI to describe muscle function and how to use these composite score and how they relate to different time tasks and muscle strength was also published the same year. Next slide, yes, please. And at last, I will present data here from the REDUX4 study, the first study using these whole body composites as pre-specified endpoints for treatment efficacy. And here we can see that what we anticipated in the beginning, that normal appearing muscles here in the graph highlighted with green do progress much more slowly than the ones highlighted in yellow, being the intermediate fast progressing muscles that have a much, much higher progression rate. And what is nice here is that we, just by using this quite simple approach on dividing and classifying muscles, we can get this really heterogeneous disease into a more nice behaving from a statistical perspective, where muscles behave much more aligned. And we see here, if we look at the responsiveness and expressed in standardized response mean, as Jeff alluded to earlier, for the clinical outcome assessments, we have here for lean muscle volume really high SRMs with over 1 and almost 1.5, depending on how long you follow it for. So we get a really, really nice efficacy biomarkers by using it in this way. And looking at the correlation to field function survive aspects, here are just some data from Redux also. This is change after 96 weeks, where we have change in MRI on the x-axis and change in time up and go on the y-axis. And we definitely see a clear correlation between MRI and clinical outcome assessments here, although it is hampered somewhat by the variability in the time function tests. And it is interesting here also that all the outliers here are data points in patients that at baseline had time up and go that were really high, so 11, 12, 13, 14 seconds. So excluding those people with less function, you get even better correlations between the and we can summarize this last slide then. So just summarizing this use of a whole body quantitative MRI is that it's definitely feasible to use in multi-site clinical trials in rare neuromuscular disorders with the rigor required for use as endpoints in clinical trials. This methodology is currently and has been in use in more than 10 clinical trials in neuromuscular disorders and used as predefined endpoints. Over 100 sites worldwide have been certified, and more than 1,500 data sets has been analyzed using these pipelines. One more thing is the quantitative MRI biomarkers of both muscle volume and muscle fat correlate to both the muscle pathology here and muscle function. And the measurements have a high analytical performance, enabling detection of really small changes in comparisons to clinical outcome measures. And these composite biomarkers have proven effective in describing muscle changes consistent with disease progression, even in heterogeneous neuromuscular diseases such as FSHD. Thank you.
Thank you, Per. That's really excellent. And I really appreciated the timeline and the progression that you showed from 2018 to now, because now as we start to think about the application of artificial intelligence and AI to not just the other functional assessments, but also to MRI, I introduced Dr. Sylvia Salinas-Blemker, who will present on a novel approach to the analysis of the identical data, and the work was done in collaboration, and she'll describe And we did receive a question in the chat about the application of AI to all of the overall data, and we'll talk about that general question after Dr. Blemker gives her presentation. Sylvia?
Thank you. Can you hear me okay?
Yes.
Yeah. Thank you for the invitation to be here, and to the other speakers. That was all fantastic. So next slide. So I wanted to take a quick step back and give you kind of a conceptual idea for how we think about the use of MRI, really. And it really comes down to thinking biologically how FSHD influences movement. It's really impaired DUX4 signaling that impacts muscle degeneration and the regeneration of muscle over time. But then if you zoom out 9 orders of magnitude, what that ultimately influences somebody's whole body function. And that's really what we care about. That Grace Fisher earlier really beautifully talked about the impact on mobility and function. Next slide. So, but the challenge is that there's a lot in between that influences the way that cellular, molecular elements influence function, from the quality of tissue to the architecture, size, properties of a muscle, to even how groups are organized. So all those complexities in the middle mean it's very hard to go from the macro to the micro without understanding what's going on between. And that's really where the MRI fits in, is explaining how a change in DUX4 or a treatment of the cellular mechanisms influences movement. Next slide. So similar to what Per nicely showed, we're you leveraging whole body MRI. There's a lot of parallels to what he described and what we're doing at Springbok. We're leveraging a whole body MRI scan. We started in the lower extremity. This is actually a research that started out of my lab at the University of Virginia. Now we've moved on to a full body analysis of all muscles, essentially from neck to feet at the individual muscle level. And we leverage an AI that we've trained and validated, and now we are excited that this product is allowed for this challenge that Seth Friedman alluded to earlier, is the time it takes to quantify every single muscle and every single slice. Now we have an AI that is able to do that. We still have a Q&A check in-house that makes sure that the output of the AI is correct, because we're always seeing new types of images, so it's always good to validate. Next slide. But that's an important, and we're kind of constantly assessing the repeatability, scan, rescan, the precision, variability, and effects of variability on our methods, which is obviously extremely important. And also, many of this have actually gone into our 510(k) clearance that we have acquired last year. Continue. Next slide. So back to the FSHD. So leveraging these whole body scans, we've really been leaning in and understanding the really full body effect of the disease and the diversity. Next slide, or next click. So we see, that's fine. Yeah, next one is good. So what we're looking at here is a lot of data, but I think it's important to have each muscle in each person deserve its own data point, because each muscle in each person is so unique. So each dot in here is a muscle in a person. And the y-axis is fat fraction, so that's the measure of disease. And in the x-axis, each of those columns is a muscle. And then we've kind of highlighted the orange dots are all up models, muscles of models, models of muscles of the upper body, and then the purple are of the lower body. So what we see is all intermixed, and it's ordered by essentially average fraction. So if you average across all of our individuals, and this is a pool of data from several natural history studies, including some Move Plus data, some data from the Wellstone project, some data from Doris Long, and actually some of the placebo data in the Fulcrum Redux 4. So the most progressed model, the most often progressed, is the semimembranosus, which is one of the hamstring muscles, all the way down. And what a few characteristics, which I think of this that I think are important to point out, or is really very few muscles are essentially preserved, because the little dashed line there is what you see in the general population in terms of fat fraction. So it's a little variable, some muscles naturally higher than others, but all very low. So very few muscles are essentially, almost no muscles are preserved across all individuals. You see dots that are high in pretty much every muscle. So this really speaks to this full body influence. But then you also see this extreme heterogeneity across muscles, and then even within muscles, like some people high, some people low. So it's just really kind of important to see that heterogeneity. Next slide. Oh, and so I also, we're advocating for, as Grace was mentioning, sort of maybe renaming or just re-talking about this disease. It's not just selectively only influencing certain muscles, as the name and the kind of diagrams that you see portraying the disease really illustrate. It's really a profoundly impactful. This next slide now shows you the kind of parallel to that, but I think it provides unique information that I think is often underappreciated. And Per was mentioning this too, and Seth too, is the impact of the disease not on only how much fat was present relative to the muscle size, but how much lean muscle is remaining, because that's really what's determining how much a muscle can function, how much force it can generate is directly related to how much contractile tissue, how much lean tissue. And so what I've plotted here, sorry, the axis got a little bit funky, but is a Z-score from essentially how many standard deviations from expected in a general population, where the dashed lines are giving you above and below a standard deviation. So as those dots, so again, it's each go below that line, it's showing which muscles are really profoundly small, so weak in this population. And so again, we see a big range across muscles and across individuals, but really illustrating. And to some degree, the muscles that are most to the right here aren't all the same ones as fat fraction. So I think it's important to understand that both the presence of fat and the loss of muscle are both important in order to fully capture this disease. Next slide. And then also there's the complexity of fat infiltration across the muscle and the distribution, which is important for being able to predict the progression, but it also influences the function of the muscle itself as well. So there's that complexity. Next slide. And so I wanted to bring this back. So the challenge is I've kind of gone wayward with how much complexity we've been measuring here. That's a lot. And, Per nicely illustrated how you simplify things to in order to make a clinical trial happen, which is really amazing leadership in developing those first fulcrum trials. So now we're saying, well, there's all this more complexity. Well, what do you do with it? Because there's, next slide or next click. There's a lot of data points, so many. So what do we do? Next slide. So our vision is to now kind of harness that and develop a digital twin of each patient that incorporates all of this information, because now we have a ton of information to create these personalized representations to essentially capture the uniqueness of the disease's impact on a particular person. So it can, the model can incorporate molecular level data, kind of regional within the muscle, this heterogeneity. It can incorporate muscle level or even group level. Next slide. And then that can be incorporated into a machine learning model, a kind of data-driven machine learning model to, next click, predict the personalized progression. And if you imagine how this would be used in a clinical trial, next click, that can then be compared to the observed change in that person, essentially providing each patient in the trial with their personalized surrogate placebo. Then you can see how they change relative to what's expected. And there are examples of these now in other diseases. I think ALS is one that's really progressed a lot. And I think MS as well. These heterogeneous diseases are all realizing this type of approach can be really powerful. So next slide. This is the approach that we've started with. We're using a random forest machine learning model that allows us to incorporate lots of different data types. And this is actually purely an example. And we have a paper that gives us lays the foundation for this. So some of the types of parameters that will go into this model for each muscle, we can get patient type of information like D4, Z4 repeat length, which will, kind of stratify patients. Muscle name is a classifier because different muscles have different types of progressions. The initial fat fraction, yes. And, as Per was saying, that, generally intermediate muscles have more likelihood to change, and that would reflect that. The other one that's come up a lot is what we call fat variation, is the fact that if a muscle has a very heterogeneous pattern or distribution of fat, it's much more likely to be in a state of change. So we can incorporate that. We can also incorporate some composite features or properties of other muscles. And then we predict a change in fat fraction or lean volume. Next slide. So this is the data that we're using. We have a lot of different data, including the actually global registry project data from that Emma Weatherly at the FSHD Global Foundation, a research foundation, has spearheaded. The Wellstone Center, Kennedy Krieger from Doris Long, Move Plus, and then Fulcrum Placebo. So we have a range of time points, up to 118 muscles per patient, over 100 unique patients, so many analyzed muscles. And so the way we design these is we use 80% of the data to train our models and then the remaining to test the prediction. Next slide. So this is just like a kind of in a nutshell, and we have more details in our paper of the performance of our model. So the training, test, and the validation, we're seeing good, strong prediction. We are aiming to add more data to this, and in particular, Fulcrum has agreed to share all of their phase 3 placebo data in here. So I think that will improve the performance. But then we can see on the right there is the performance once we look at the group. So on the left, each dot is a muscle on a person prediction. And then on the right here is each dot is a group of muscles in a person. So as we group muscles, they definitely, some of the noise associated with the predictions starts to go away and it gets very tight. Next slide. So this is just not to intend to see all of them, but showing that we can do this prediction across all the muscle groups across the body, which are relevant. On the top, we have prediction of fat fraction change. And on the bottom, we have prediction of lean volume change. So both of those, as I was talking about, are very important for predicting function. Next slide. And so I wanted to go back to this intermediate muscle concept because I think it's really important. And the vision that Fulcrum and Emmer had in terms of saying, okay, well, in order to address the disease, we need to kind of pick out the muscles that are most likely to change. And so what we're looking at here is the top is the plot of the change in fat fraction, and the x-axis is the initial fat fraction. So the highlighted red dots are each muscle, and where those are the intermediate muscles, these in the kind of quote unquote B muscles, the intermediate. And so the higher that number, the higher on the y-axis, that means they're more progressing. So those are the muscles that are most progressing in these particular patients in the natural history sense. And then the y-axis similarly, so fat fraction is going up, lean volume going down, kind of as the disease progresses. So what we see there is, yes, many of the high dots are red. So yes, the intermediate is capturing muscles that are changing a lot, but it also incorporates many muscles that aren't. So the initial state of the muscle in terms of fat fraction is an indicator of that it may likely change, but it's also it's not really filtering out muscles that aren't. And also there's muscles that are changing. In particular, if you look at lean volume down there, that aren't in the blue. So if you go to now click on, So this is now a new version of that. So now we've used our model prediction to say which, and now what I've color coded there is the model prediction of change. So where red is high prediction of change, and then blue is low prediction of change. And so that we've mapped that onto these same points. So now we see that this model is discerning across the ranges of fat fraction which muscles are most likely to change, and versus the ones that are blue are all not likely to change. So this is showing you essentially it's an equivalent idea that the original team had in terms of isolating which muscles are going to change, but in a now kind of on overdrive where we can capture all sorts of elements of the muscle and the individual to say what's likely going to change. Next slide. And then finally, I wanted to back to function. We've been doing a lot of data-driven modeling of relating the muscle properties with functional tasks, and I don't want to belabor many of them, but I wanted to go to reachable workspace because I know it's of great interest. And so this is, and we've done this for baseline muscle properties versus baseline reachable workspace. We've also done this with change. What I wanted to show you, so we can do a training and a testing, and these models perform quite nicely. What I wanted to point your attention to on the right is the model also gives you which muscles or here muscle groups are most influential. So the further out from zero those dots are, the more influential. And it's showing you essentially as you change the fat fraction, it affects the reachable workspace. So what it's showing is certain muscle groups are much more influential than others, which is biomechanically makes sense. But I think this is a nice data-driven illustration. So if you're having somebody who's changing in those particular muscle groups, it would stand to reason that that person would also likely change in reachable workspace versus others where those dots, those particular muscles are more the blue dots than the red dots, then it also wouldn't stand to reason that their reachable workspace wouldn't change as much. So next slide. So just some final points. These MRI measures provide this critical in-between information to essentially help target the clinical trials, help address the heterogeneity across the individuals, and really explain what's happening, and really clarify that relationship between the treatment and the patient function. With that, I'd like to acknowledge our collaborators and our funding for this work. Thank you.
Thank you, Sylvia. Thank you, Per. Those were really very detailed, excellent approaches to thinking about how we engage the MRI data. I'm going to actually go back to Seth with the remaining 2 questions. We've used a lot of our time for the MRI in our discussions, but I think it was so relevant and important to be able to look through that data. Seth, I want to ask you how other imaging modalities compare for sensitivity and reliability to MRI, and using FSHD as an example. Or
Yeah, thank you, Lucienne. I think the biggest challenge is that a lot of the data that I've seen in the literature, if you measure a single slice of anything, the reliability is pretty good. And I think the dilemma is that the disease is very heterogeneous in terms of its, expression in that muscle. And so the likelihood that you'll either miss the area that is injured, or you'll miss the change that is occurring over time is very high. Certainly with modalities that pick individual regions, I think, ultrasound is oftentimes it's very inexpensive. I know it's like Larry Hayward could probably comment on his experience with it. But it is a little finicky in terms of how the acquisition and the analysis is done. And so I think we've really been trying to move towards something that is, covering the whole body seems very important in terms of evaluating, is the trunk okay? Is the shoulder girdle okay? Which muscles are involved? And, the only modality that really is sort of whole body, DEXA, has been utilized in some studies, and that certainly is a very inexpensive way to get an idea of body composition. I think that it's going to be very difficult to link DEXA back onto function. And as Sylvia mentioned, I mean, I guess what I would love to see is this, I think there was a word that was mentioned, sort of this functional progression, sort of meaningful progression. Like, there really does seem in any sample only so many subjects that will likely have functional progression in reachable workspace over a study trial. A different group of subjects in that cohort may have change in their walking function. And so really to be able to pick out those subgroups and take the explanatory variance that you get on the imaging and map it onto the explanatory variance that makes sense from the functional tasks seems like the way those are going to knit together. The one caveat I would say even about MRI is that we absolutely can see change in MRI that will not have functional consequence on the tasks. And I think, as Sylvia mentioned, a lot of times you have to go towards these muscle assemblies, things like, adduction, to get a sense of what the function is going to be. And so it's, I think, very useful to imagine that we're going to have different time scales, that the MRI is probably going to change faster than the functional outcome measure, but you can probably use the MRI to inform which functional outcome measure will be a good reporter.
Yeah, thank you. That's really clear. And it's amazing how much, even with a residual amount of muscle, you could still retain some function. Then I have one other question for you, Seth. I think you've got the most experience here, but what are the challenges of evaluating a complex population like FSHD, where we have young, we have rapid progressors, we have later, slower progressors, we have ambulatory and non-ambulatory. So is MRI evaluable across all of that population? What are the challenges there?
I mean, I'm wearing my pro MRI t-shirt, if you didn't see it. No, I mean, I think that, this is one of the reasons that we're really excited about starting this NIH-funded pediatric study. Like, I think that there are definitely different challenges in using MRI in pediatrics that mainly relate to the analysis. We really have to understand how to correct for normative growth. And the other piece is that, as I mentioned, I think what Sylvia and her team have really discovered is that the quality of the bone, bone volume really can constrain muscle changes. And so in a child, if you have, if bone is abnormal, it may really change how the muscles grow. So there's some real complexities in the pediatric sample that I don't think we fully, we have enough data to really solve. I think to the point that, Jeff brought up with this question of non-ambulatory, I think MRI is certainly very good at evaluating the whole body. In a non-ambulatory patient, you're just going to need a different functional outcome measure to assess if the drug is working. And so I think this real understanding, and I know that, Sylvia, as a biomechanical engineer, we've had a lot of discussions about this. Really, we need to know for each task, which is kind of the set of muscles that weights most highly onto that function. And then I think, to Grace's point and the points that Jehan raised and others, is like, patients don't move the same with FSHD as normal subjects. And so, one of the things I think we've been really excited about these computerized tasks and other types of tasks is you might be able to map the quality of motion onto the MRI data, because we have so much granular data now that I think that's an exciting opportunity to use, sort of more unsupervised methods to really try and kind of put those two things together.
Thank you so much, Seth. So that was a really informative session on MRI, and it's time for us to move to molecular biomarkers. And Dr. Suja Jagannathan, who's assistant professor in Colorado, will speak to us about what's been happening in the FSHD space.
Yeah, thank you, Lucian. It's really an honor to represent the community and talk about what kind of FSHD biomarkers have been developed that are in the process of being developed. So I'm excited to talk to you about it. Next slide, please. Okay, so as we have heard today, we have heard about, clinical markers, functional testing, imaging markers, and my talk is really going to focus on molecular markers, including things we can measure from the biopsy or the blood. Next, please. So I just want to give a very broad overview of the markers that I'm going to touch on today. So obviously, depending on what the goal is, you can have different types of biomarkers to track a disease, to stratify patients, to monitor if someone's improving or not, and so on. So next, please. For FSHD, for the longest time, we have worked on having a good diagnostic biomarker, right? That was a barrier not that we have now, I would say, overcome. And that bodes well for the next barrier that we are now facing, which is to find a really good response biomarker that is going to help us ask if a drug or therapy is actually effective in treating a disease. And that's the biomarker that I'm going to focus on today. Next, please. All right, so what does an ideal response or pharmacodynamic biomarker look like for FSHD in particular, right? We would like something that is present in the diseased muscle or preferably in circulation, something that correlates with DUX4 levels or activity because that is the main disease driving mechanism. Something that can be reliably detected, right? That's a big barrier too, given how heterogeneous and mosaic DUX4 expression is. Being able to see something reliably over time is an important aspect. And finally, and probably most importantly, what we observe needs to correlate with disease severity or the response that the patient has to treatment. In other words, it needs to actually benefit the patient in terms of what we are looking to track. All right, next, please. So there's sort of three main classes of biomarkers that I'm hoping to cover today, just to give everyone a broad overview. There's, we can track muscle status itself. Now, this is not specific to FSHD, but just asking how healthy is the muscle in someone, right? There's also a lot of work being done in inflammation-related biomarkers, given how important inflammation seems to be for FSHD pathophysiology. And finally, the directly DUX4 regulated biomarkers that I will also touch on. Next, please. Okay, so for muscle status, these studies go back decades now where people have tracked things like creatine kinase, which are enzymes that are released into the blood when muscles deteriorate, right? So this is a very physical marker of asking, is there muscle damage in a person over time? And this is a blood-based biomarker, and it's been done in multiple cohorts and natural history studies now, and shows that in people with FSHD, you do see an increase in many of these muscle markers. Now, the downside to these types of markers is A, it is not specific to FSHD. There could be confounding variables, right? If somebody did an extreme bout of exercise, you're also going to see change in these types of biomarkers. So there are pros and cons to it, and that is something to keep in mind. Next, please. Next slide, please.
Sorry, thank you.
Yeah. So the next type of biomarkers, right, the inflammation. So this has also been a really active area of research in FSHD because we know FSHD affected muscles have inflammation happening. And the example I've chosen to highlight here is interleukin-6, which when you measure it in controls and patients, you can clearly see that there is a separation between these groups, right? And this has now been done in multiple studies, and there's definitely a consistent increase in inflammatory cytokines, not just IL-6, but also others that you can track in the serum of a patient. Again, the downside to this is, as you can see, there is a pretty good overlap in the signal between controls and patients, meaning you cannot just pick out one individual and be able to confidently classify them into, if they're affected or not. But over time, if you have a baseline measurement, this could be a really good way of tracking what's going on with inflammation in that individual and are they responding to treatment, for example. Next slide, please. And another type of inflammation-related biomarker too is the complement components that are present in the blood. And this is a composite of 4 different complement markers that were measured in this particular cohort. And again, you can see that there is separation between control and FSHD. The same downside of the inflammatory, the interleukin biomarker applies here in the sense that you have a pretty significant overlap between the 2 populations, meaning we need to better understand in a longitudinal manner, how does this change over time in someone with disease? What is the natural variability of the marker in someone that is healthy, right? Like if you have an infection, are some of these things going to shoot up? And how do we account for those confounding variables when we do an actual clinical trial? Okay, next, please. All right, the last class of markers that I want to focus on is DUX4 regulated. Now, this has been sort of probably the most widely adapted in clinical trials that have happened so far. So what you're looking at here is RNA sequencing done on muscle biopsies. And then we are looking at a composite score of 4 different DUX4 targets and their expression level across individuals with and without disease. And what you're looking at here is that there is a correlation, a strong correlation between individuals with disease and higher severity and how much of this DUX4-induced transcripts they have. The biggest caveat to this approach, and next slide, please, is the reliability of biopsy material, right? How well you, not even well, like it's qualitative, right? You choose a site to biopsy and whether or not disease is active in that location, whether or not DUX4 is being actively expressed in that place and in the material you collect could have a huge impact on being able to confidently assess this biomarker. So for example, in the REDUX trial, there were, sort of improvements in some functional testing in people that took the losmapimod, right? To the left, you're looking at their ability to lift small amounts of weight. And there was definitely in individuals that took the drug, there was marginal gains in this particular measurement. At the same time, when you look at the DUX4-driven gene expression, there was no change that was statistically significant, right? So what this told us is that you can have perhaps some meaningful change in an individual, but it may be too small. And depending on the biopsy location and how the study is done, it may not be a sensitive enough measure to be able to track disease, right? And that is the problem in some ways with this measurement, because you're trading sensitivity for specificity. The more specific you try to be in a biomarker, it could also be the less sensitive. And that is something to weigh as we evaluate biomarkers that come up in how we handle them. Next slide, please. But I also want to say that, this is not consistent, right? This is another trial that also used the DUX4 gene signature with the biopsy study. And this is the Fortitude trial, where they clearly see a reduction in DUX4 score as patients get treated by the Delbrax. So again, this is just highlights that there is variability and subjectivity in how we handle sort of biopsy-based biomarkers looking for DUX4 targets. And that is something to keep in mind as we design future studies and how we balance the risk of getting the right measurement with other biomarkers that might give you other measures that we may want to use as a composite, right, to track disease. Next slide, please. Okay, so I want to also touch upon a few biomarkers that are sort of up and coming that we should be on the lookout for as they become widely adapted and vetted by the community. So one example is the next slide, please. The marker that was reported as part of the Avidity trial. So they reported a novel DUX4 regulated biomarker that they could detect in the plasma. We don't know the identity of it, but it definitely from the data looks like it's something that tracks with dosing of this drug and as time progresses. So that is something to look out for once we know the identity of it and it's adapted by other studies and in multiple sites. We'll be able to know how well this functions. Next slide, please. Another one that's also under active development is the SLC34A2, which is a DUX4-induced protein. And again, something that can be tracked in muscle itself, which we cannot visualize DUX4 on its own in muscle. So this can be powerful. And similarly, looking for this in plasma. And there's also biomarkers that I'm not talking about today that are under active development. And it's just a matter of adoption by the community and vetting and peer review. So for example, my lab is developing a biomarker that looks at cell-free DNA in the blood and asking if could that be a way to track presence of disease. And these are biomarkers that are coming up. So I would just say that this is an active area of research where a lot of labs are putting in important work, and it is a matter of time before hopefully we'll have something the community can as a whole agree on that tracks disease and tracks how well a patient is responding to a treatment. Next slide, please. I would like to finish with these thoughts, which is, so how do we evaluate these biomarkers and how do we move forward in this? It's obviously a huge unmet need in the FSHD community, given how variable the disease presents. The attraction with the molecular biomarker is by simply tapping into the bloodstream, you might get a readout that is a whole measure of everything that's happening in the body. And that is what's attractive about a molecular biomarker. And it also moves us away from, the subjectivity of who's doing this, the imaging or how it's being interpreted, to something that is just a molecule you're measuring in a clinical lab, right? That helps really. But the things to consider are, of course, sample availability and preparation. We heard from Grace earlier, muscle is the precious thing we are trying to protect here. And to have to take chunks of muscles to evaluate if the disease is happening, that comes at a cost. So thinking about the burden on the patient and how can we make a measure that is the least burdensome, but still something that is meaningful in tracking disease is an important consideration. And another point I want to touch on is also endpoint duration. So what I mean by that is, if somebody is actually improving with a particular therapeutic they're on, how quickly is the biomarker going to reflect that, right? If your biomarker is very long, it takes a long time in having a lag, you may miss things or you may not be doing the trial long enough to see a meaningful change. If your biomarker is too rapid, then you're going to have daily fluctuations. So depending on the day they show up in the clinic, you might have variability that you don't want, right? So that is something to evaluate in every biomarker that comes up. And that requires longitudinal studies that pair the biomarker with other imaging markers, MRI and functional testing, to give us a better understanding of how do each of these molecular biomarkers compare and track with various other metrics that we are measuring, right? And finally, given the sort of slightly later onset in individuals and just variability in how quickly the disease progresses, there are many confounding factors to consider. For example, the age of an individual can have a huge impact on the biomarker you look at. The sex of an individual, did they have a cold recently in the case of inflammation, right? Like these are all things that we do not know yet, but are going to be super critical for us to evaluate again in future studies to be able to confidently use biomarkers as a way to track disease. And again, this is active area of research. Many labs, including ours, are working on this. So I'm very hopeful for what lays ahead. But I would like to finish with these thoughts.
Thank you very much, Suja. I'm going to stop the slides and we'll have some discussion. So Dr. Leo Wang is available to us by phone, and he's been deeply involved in the biomarker arena as well as the clinical side of evaluating FSHD. And I want to ask Leo, are you there? Can you hear us?
I can hear you. Can you hear me?
Yes, thank you. Great. So can you give us your sense, your perspective on how the community has sought to correlate the amount of and the change of these molecular markers, or you could discuss MRI if you wish to, with PROs or with functional assessments, and include in your answer to what extent these markers have been assessed and validated in independent patient sample sets?
Right. Thank you so much for having me. I apologize that I'm not on. I'm calling in. I'm traveling, and this is the best way for me to participate.
We appreciate it.
I'm a clinician. I'm a professor of neurology at the University of Washington, and I see patients as well as participate in natural history studies as well as the clinical trials. And I'm going to speak from perspective of somebody who's participated in this gamut of patient-related research. But first of all, I think that what we've learned is that MRI can inform us with the molecular target, the molecular biomarkers, and understanding which muscles to biopsy and which muscles may have activity that will give us a great sense of the activity of the muscle specifically, or the activity of DUX4 that's turning on potentially this toxic program and this deterioration of the muscles. With that, we have looked at terms of correlating the changes in the muscle biomarkers to how the changes are in the MRI, and seeing how MRI, specifically of specific muscles, how they change over time. And you can see some correlation in that there is some activity of DUX4 that can correlate with the changes in MRI, but it is a marker that, like, similar to what has been described in the last few hours, is just there's a lot of variability. And I think that variability is due to quite a few things. One is that, we've talked about within a muscle, there's a lot of changes, a lot of differences within it, that it's not even just a proximal distal gradient, but there could be islands where DUX4 could be turned on and then it spreads. And so there's something that you have to take into account. Second, I think that when you have multiple sites, you want to really be careful about how you approach the biopsy and try to make sure that you biopsy a region that is important. And so, and then finally is the method by which you analyze the muscle biopsy samples to look at the molecular biomarkers. I think that those three things really really determine the variability of your muscle molecular biomarker. And you can see that from the natural history study with Wellstone and other international studies, with the study with losmapimod, and now with the study with avidity, there seems to be some improvement and some hints that this molecular biomarker, our ability to assess that, has really improved over time. We have looked at these really mainly in how the changes are with the MRI. It's a little bit tougher to determine the relationship with patient-related outcome measures at the patient level, just because we're really sampling one spot of one muscle that is part of a really heterogeneous disease process. I think I'll stop there and let for further discussion.
Thank you, Leo. We did have a question that I'll ask if you would like to address, or given your experience, or if Jeff would address it. Has there been research done on the effect of hormones on muscles? We've talked about bone density, and it made one FDA member think about menopause and women losing bone density and muscle due to loss of estrogen. So I know there's been a good amount of work done on hormonal impact of and correlations with severity and progression of disease. So Leo, do you want to make some comments? And Jeff is here as well.
That's an area that I'm not as aware of, so I'll probably let Jeff speak more about it. But I think that there has been some studies. I'm looking at growth hormone, human growth hormone, as well as testosterone, and there's some hint that there is some increase in muscle by the DEXA scan, and so further studies are required. But that's probably, I think, the one study that has a little bit more rigorousness to it in terms of understanding the interaction of the hormonal system in FSHD disease process.
Thanks, Leo. So Jeff, I'll ask you to comment here.
Yeah, I'm happy to. I think some of the other people on also would be qualified to address this. I think in the field, there's been a lot of interest in the, sex hormones or effects of estrogen on DUX4 regulation, because there's some differences in the age at which men and women are diagnosed with FSHD. This is thought that it might be hormonal. There's some studies suggesting a role for estrogen in the trafficking of DUX4 in the cells. No actual trials other than some retrospective studies seeing if hormone replacement may have affected individuals, but they were unable to determine any real effect. Then Leo's right. There was a prospective study of testosterone and recombinant growth hormone. This was thought to be an anabolic approach to increase muscle growth. And we're able to see that, but in an open label study. So really needs to have some follow-up. And I know the researcher who did that study is actively working to make that happen.
I would also add, I mean, we could take, I know there's minimal studies on menopause and changes in hormones related to disease, but when we look at menopause and sarcopenia, we know that estrogen does have an influence on satellite cells that'll be responsible for the regeneration and repair of some of these muscles, as well as an impact on inflammation of muscle tissue. So I think some of those things are worth exploring and learning from other areas that had more research on it.
Thank you, Jeff. Thank you, Tina. And Nizar Saad has joined us today, and he, together with Suja, actually are pushing the forefront of our understanding and our discovery of biomarkers. Nizar, would you introduce yourself and speak a little bit about the future coming areas regarding molecular biomarkers?
Yeah, sure. Thank you, Lucienne. I think, yeah, I mean, Suja and Leo touched base and commented about the biomarkers and the importance of having molecular biomarkers for FSHD. And I think the most important area to investigate and keep looking at is mostly the circulating biomarkers, molecular biomarkers, because they will cover the whole body effect of the disease progression. And so some of the new areas that should be focused on, or actually I'm actively working on, is molecular biomarkers that we could track and know that are coming from skeletal muscles, and that you can, as possible, be able to detect in circulation. Two things that has to be focused on definitely is how variable those biomarkers will be and how sensitive the detection method would be. With the sporadic expression of DUX4, you might need to have a very high sensitive method of detection, but also reducing the variability as with the other outcome measures that people are using or preparing to use in clinical trials. So, but I think as the clinicians are preparing all these outcome measures to reduce as much as possible the variability, we have a good chance of identifying some circulating biomarkers that could also be less variable but highly sensitive in detecting change and progression in FSHD.
And I want to go back to Suja's slide that showed, like, what do we need to in a molecular biomarker or in an imaging biomarker? But I think Sylvia addressed and that whole team addressed that very nicely. So in a molecular biomarker, what do we need to do to really understand how well it really reflects disease?
I can take that. Yeah, I think one of the things to understand is like Nizar said, the source of it, right? Because it could be that, we are looking for things that are coming from muscle, but we could also look for things that come from the immune cell population that's actually responding to DUX4, right? There are many ways to meaningfully track the disease. And I think as we do studies on molecular biomarkers, still having a sense of what is the source of it, how does it relate to disease, I think is quite important. And then thinking about, again, how variable is it in a healthy individual versus in an individual with disease? How quickly does it track? You could lose a muscle function overnight, right? But probably the actual process of that muscle being lost happened over time. How quickly do you need to be able to measure a biomarker to be able to see the change? How persistent is that signal in the blood, right? All of those factors are going to be important.
Wonderful, thank you. And I wonder if there are any other short comments any of the participants would like to make before we move to a summary that I've prepared. Any other comments or points that you'd like to raise? One thing I'd like to raise based on what Suja just said is that there is immune mapping going on of this disease in natural history studies, and we've given very limited time and only shown a short and small glimpse into our understanding of how the immune system contributes to the disease. So there's very clear immune infiltration of the muscle. There's very clear circulating proteins responsible for activating the immune system, and much more will be known about this over the course of a few years as we map adults and children, pediatrics, with by very careful mapping that's happening in Australia, in the United States, and in Europe. So to summarize what I'll just speak to one question that we had, and it's around patients and how interested are patients in being involved in the clinical studies. And I'm going to address that in my summary, but Grace Fisher has also raised a useful and valuable point that the society has heard from many patients, and that's the challenge of being involved in clinical trials. The challenge of the travel, and when somebody is challenged with mobility, they're challenged to get to a clinical site when they have jobs, and they're challenged to get to a site. But there's very strong interest to be involved, and having come from industry and now working with a society, that is very clear with the numbers of biopsies that patients are asked to do, and they've met that requirement, the numbers of MRIs, the functional endpoints. We heard from Grace, it's very clear. There's exhaustion, fatigue associated with carrying out those extensive analyses that occur in clinical trial settings. And so education, I think, of trial sponsors and education of patients is important. And that's one gap that the society hopes to work to fill. Some disease areas have actually released, recommendations to trial sponsors, and I think that's something that we could work on in the future. And giving logistical assistance to patients as they seek to be involved in clinical trials is going to be very important. So over the course of this couple of hours, we heard how relentless FSHD is in its progression of muscle wasting. The muscle loss is quixotic, as you've seen very clearly from the MRI, and it leaves patients and their families in a state of constant dread, and also in mourning for what they are repeatedly losing over time. Larry Hayward, when he was talking with us in one of our early discussions, was describing this very strong individual with FSHD who came in with all these beautiful muscles, but for some reason he'd had some sensation and some rippling in one of his pectoral muscles, and then in the course of a few days he described to Larry how it was gone. And it's to visualize that type of loss of something that's essential for your functioning as a human being is just so challenging. So it's repeated loss, and weakness and pain are broadly experienced by people with FSHD. Also, respiratory insufficiency, as you heard from Jeff, may shorten the lifespan, and it certainly impacts the daily living of certain individuals with FSHD. What do we know about the patients? We know patients are supportive of treatments. They want treatments that will either slow or halt the progression of the disease. FSHD patients welcome restorative treatments on top of that to improve muscle strength. As we halt the activity or the effect of DUX4 in muscles, how do we help patients rebuild muscle? Patients with FSHD, as I've said, are clearly eager to work with drug developers and regulators. They provide biopsies, they undergo the MRIs, and they enlist in patient registries and provide a lot of information about their lives and about their personal functions. But they realize it's essential as we work together to expedite the approval of new and safe, effective therapies. So currently, very clearly, there's no FDA-approved treatment for FSHD that prevents or slows the disease. And we're very fortunate, as you saw in some of the slides, to have a deep therapeutic pipeline. You also could see the value and the promise of AI to allow us to analyze these really deep and complex data sets to allow us to arrive at which are the markers that change and how do they change in concert and what can we measure in a short time to really evaluate the disease. So as we conclude the educational session, the Society really sincerely thanks all who have contributed to the development of the outcome measures we discussed today. First, of course, we thank patients and their families who have provided essential input on the disease. We also thank the clinicians who have driven the study of the natural history of FSHD. Jeff Statlin, Rabi Tawil were really the founders of the concept of the Clinical Trial Research Network, have done really foundational analyses there. And there are European sites, there are European centers. So, and Asia and India are also very doing a lot of intense work for identification of patients, genetic analysis of those patients, and we're seeing similarities and we're seeing differences as we look at worldwide populations. Also, there are studies in Brazil and there are recent studies in Northern Africa. So truly, with that genetic analysis being very approachable, that's our understanding and our ability to find patients and then help patients globally is really enhancing. The scientists who uncovered those genetic mechanisms, because it's very complex genetic regulation that drive DUX4 expression, we want to thank them as well. And the scientists who characterize the FSHD muscle and lead us to an understanding that DUX4 is a transcription factor. It turns on a lot of other factors, and no one of those hundreds of proteins is responsible for the disease. But there's this multitude of transcriptional activity and repression that's driven by DUX4, and that altogether is really driving the disease. And we also heard that DUX4 expression itself is quixotic and it's challenging to detect in a reliable manner in muscle. So these secondary endpoints, the DUX4 regulated transcription proteins, are really essential for our ability to track DUX4 activity in any kind of molecular way, and demonstrating the relationship between, in our preclinical studies, because it's so challenging in patients, but preclinically of a treatment on DUX4 levels together with DUX4 regulated transcription proteins can help us develop a understanding and a validated approach to thinking about surrogate markers of DUX4 activity. The unregulated immune activation in the form of interleukins and complement was described by Suja, and there's a lot more to be discussed there. So finally, I'm going to answer the question that I asked earlier, which is if a therapeutic benefit is seen in a clinical trial population, is it reasonable to extrapolate to the entire FSHD population? And I got a very thoughtful answer from Jun Kinoshita, who's worked with the Society for so many years and has been so essential in supporting patients for more than 20 years. If the therapy stops DUX4 expression and a benefit is seen, it's reasonable to expect other patients will benefit. The reasons are that we know that the same disease mechanism is present in all patients and that these patients progress in their symptoms. It's relentlessly progressive disease. Someone who's too mildly affected to be included in a trial will inevitably worsen over time. Likewise, someone who is too severely affected, for example, by being non-ambulatory, will also progress and lose upper body function, will lose core function, will lose breathing functions, and will decrease their quality of life and ultimately their lifespan. It's therefore reasonable to expect that patients will receive clinical benefit even if their condition did not allow their functional changes to be measured by the outcome assessments used in a trial. There is no evidence that people with FSHD have only a limited window in which they would benefit from a DUX4 targeting therapy. So even if they have profound loss of muscle, there is still reason to believe that they could benefit by allowing them to retain the muscle that they have. As Sylvia made clear, you could be down to very small amounts of muscle and you've still retained a component of function. So I just want to end by thanking everyone again. And we appreciate the questions that came to us from the FDA, and we appreciate the questions you sent to us ahead of the meeting. And we look forward to more fruitful interactions.